Hello friends, so this is the third disease we are going to discuss, the bacterial leaf blight of rice. We have already discussed about the major rice diseases, that is rice blast and brown spot. So here similarly three section will be there, MCQ, second section will be the SSR reasoning type, and third section will be quick revision points. So this will be helpful mainly for the ARS net as well as for the SRF exam, JRF exams also, and other competitive exams also. So let's start the video. So the question number 1, the bacterial leaf blight of rice is caused by, very easy question. So option are Pseudomonas syringae, option B Xanthomonas oryzae pathober oryzae, option C Xanthomonas oryzae pathober oryzae cola, and option D Erwinia caratovora. So the answer is Xanthomonas oryzae. And if the pathober is oryzae cola, then it will cause the bacterial leaf strip of rice. Okay, BLS. This is BLB and that was BLS. Then question number 2, the BLB or bacterial leaf blight was first discovered in. The options are A China, B Japan, C Philippines, and D India. So the bacterial leaf blight was first discovered in the year 1884 in, it is first discovered from Japan. Okay, remember this is important question. First discovery with year you should remember. Next, question number 3, Kresek disease was first reported from. Options are India, option B Indonesia, option C Thailand, and option D Japan. So the Kresek symptom, this is the sudden wilting at nursery stage, generally 3 to 4 weeks seedling. So at that stage sudden wilting occur due to the infection of bacterial leaf blight, and this was first reported in the year 1950 from which country? From Indonesia. Okay, so Kresek disease, this is stage of BLB which is first reported from Indonesia, but the BLB itself was first recorded from Japan. Next, question number 4, a severe epidemic of bacterial leaf blight in India was occurred in. So here the options are option A 1950, option B 1959, option C 1963, and option D 1970. So severe epidemic in India occurred in the year 1963. Next, question number 5, the characteristic symptom of BLB is. Option A water soaked lesion progressing from leaf tip. Option B brown spindle lesion. Option C small balls. Option D rust pustule. Here the answer is water soaked lesion progressing from the tip. So what happened in the BLB? It is the leaf. So water soaked lesions start from the margin and tip. Okay, this will further grow both length and width wise, and it will grow into straw or yellow color strips, yellow color lesions with wavy margin. This is the difference between bacterial leaf blight and bacterial leaf strip. In the bacterial leaf streak, it will form the streaks, no wavy margin is there, but in case of bacterial leaf blight, the margin of the lesions are wavy. Okay, wavy margin is there. Next, question number 6, Kresek phase is characterized by. Option A local lesions, option B systemic wilting of seedling, option C root galling, option D grain discoloration. Here the answer is systemic wilting of seedling. So Kresek phase generally occurs at 3 to 4 week seedling at the nursery stage. Sudden systemic wilting occur, that is known as Kresek, which is first reported from Indonesia in 1950. Next, question number 7, the pathogen primarily enters through. Option A hydrothores and wounds, option B roots, option C flowers, option D panicle. This is a very important question and most frequently asked question in most of the entrance exams. means state exams, other competitive exam, as well as in the JRF, SRF or ASRB exams. This is most frequent, as you have to remember this. Okay, Haida Thors. Then question number 8. Godavari district of Andhra Pradesh in India is endemic to option A BLB, option B BLS, option C rice blast, option C sea throat. So the Godavari district of Andhra Pradesh, this is endemic to BLB. Endemic means this diseases occur in very high intensity in a particular area, restricted area. Okay, this is in a very severe condition. Next, question number 9. The causal bacterium is option A gram positive, option B gram negative, option C acid fast, option D cellulose. So the causal bacterium, that is the Xanthomonas oryzae pathober oryzae. This is a gram negative bacteria. It's aerobic in nature, then non-spore forming, and it is capsulated, means capsule is there. Okay, this is the characteristic of the Xanthomonas oryzae pathober oryzae, which is causing the bacterial leaf blight of rice. Next, question number 10. The flagellation of the pathogen is option A peritrichus, option B amphitrichus, option C monotrichus, option D lophotrichus. So here the answer is monotrichus. Monotrichus means a single flagellum will be there. Peritrichus means all around, all the peripheral area, means entire perimeter. This is circle, means this perimeter, total area is the perimeter. All over the bacterial cell, the flagella is present, that is known as peritrichus. Amphitrichus means at the both end one flagella will be there. Lophotrichus means at both end bunch of flagella will be there. And another term is there cephalotrichus, means it is just opposite of the lophotrichus, means at one end bunch of flagella will be there. This is the classification of flagella. But in case of Xanthomonas oryzae pathober oryzae, the causal agent of BLB, it has monotrichous flagella. Next, question number 11. The pathogen colonizes mainly. So option A cortex, option B xylem vessel, option C phloem, and option D epidermis. So here the answer is xylem vessel. Generally, we thought that systemic disease means the pathogen will restricted to the phloem tissue, but here the pathogen generally colonizes the xylem vessel. Okay, this is a systemic pathogen. Initially it will enter through hydathodes, then it will colonize the xylem, and it will spread systematically throughout the plant and causing the wilting symptom. Next, question number 12. Bacterial ooze consists of option A spores, option B mass of bacterial cell, option C nematode, option D virion. This is very easy question. Mass of bacterial cells. Next, question number 13. Favorable temperature for BLB development is. So the option are 10 to 15 degrees Celsius, option B 15 to 20 degrees Celsius, option C 25 to 30 degrees Celsius, and option D more than 40 degrees Celsius. Here the answer is 25 to 30 degrees Celsius. Next, question number 14. Which factor strongly favors BLB epidemics? Option A drought, option B heavy rainfall and wind, option C frost, option D low humidity. So here the answer is heavy rainfall and wind. So this is actually responsible for the secondary spread of the bacterial cells. That's why that is leading to the BLB epidemic. Next, question number 15. Excessive application of which nutrient increase disease severity? So the options are A potassium, B zinc, C nitrogen, D silicon. So here the answer is nitrogen. As we all know that the excessive application of nitrogen leads to excessive vegetative growth, which is favor various diseases, various foliar diseases. Next, question number 16. The clipping inoculation technique was developed primarily for option A screening of resistance, B seed treatments, C forecasting, D. Studies, storage studies. So here the answer is screening of resistance. So generally for the BLB particularly, the pathogen is inoculated by clipping the tip of the leaf. So they will clip the tip and they will dip the tip portion, wounded tip portion, in the bacterial suspension for screening of the resistance varieties. Next, question number 17. Which resistance gene is widely used against BLB? Option A, XA21. Option B, LR34. Option C, SR31. And option D, PI1. So, answer is option A, XA20. This is a widely used BLB resistance gene. LR means this is leaf rust resistance, SR means stem rust resistance, and PI, this is a rice blast resistance gene. Next, option 18, question number 18. Which of the following is a recessive resistance gene? Option A, XA4, option B, XA5, option C, XA21, and option D, XA7. So, XA5, this is a recessive resistance gene for bacterial leaf blight of rice. Next, question number 19. Gene pyramiding in BLB aims to: option A, increase fertilizer use. Option B, broaden resistance spectrum. Option C, increase bacterial growth. And option D, reduce tillering. Here the answer is it broaden the resistance spectrum. Means gene pyramiding means multiple, we will incorporate or we will take multiple resistance gene in a single variety through a repetitive back crossing method. Okay, so that will broaden the resistance spectrum, because if in a single variety multiple resistance gene will be present, then it will be so resistant to multiple races or multiple strains of the pathogen. Next, question number 20. Pathotypes are important because they: option A, overcome resistance. B, increase seed size. Option C, improve yield. Option D, reduce virulence. Answer is pathotype are important because it overcome the resistance genes. So pathotype is a group of pathogen that can overcome a particular resistance gene. Means, so before that the pathogen is showing susceptible, means that resistance gene is working particularly, and it can inhibit the pathogen growth, but a new pathotype is actually generated which can overcome the particular resistance gene. Next, the question number 21. Which virulence factor is associated with Xanthomonas species? Question number answer, option number A, xanthan gum. Option B, chitin. Option C, ergosterol. And option D, melanin. So here the answer is xanthan gum. Xanthan gum is nothing but it is the EPS, exopolysaccharide, that is present around this bacterial capsule. So this is actually the virulence factor. It clogged the xylem vessel. Ultimately, the wilting symptom we observe. Then this chitin, this is present in the cell wall of the fungal cell. Then this ergosterol, this is also present in the fungal cell. Then melanin, this is important factor that is present in the aprisodium, that is helpful for the pressure buildup in the aprisodium, that is produced by, I have already told, the oryzae, that is also produce the pressure back, or the aprisodium, that is helpful for the penetrating the leaf surface. Next, question number 22. Tal effectors are important because they: option number A, manipulate host gene expression. Option B, fix nitrogen. Option C, enhance colony formation. Option D, induce resistance. So TAL means transcription activator like effectors, means it will manipulate the host gene in favor of the pathogen. So here the answer is it manipulates the host gene expression. Next, question number 23. The best long-term management strategy is: option A, resistance cultivar. Option B, continuous antibiotics. Option C, deep plowing. Option D, heavy irrigation. Here the answer is use of resistant cultivar. This is for the long term strategy. Okay, for most of the disease where this gene interaction has been established, you can go for the resistant variety. Next, which chemical is commonly recommended against BLV? Option A, streptocycline plus copper oxychloride. Option B, mancozeb. Option C, oreofungin. And option D, tricyclazole. Here the answer is streptocycline plus copper oxychloride. This is the best combination for the management of BLV. Tricyclazole, this is for the management of rice blast. Mancozeb mostly used for the foliar diseases. Oreofungin, this is a antifungal antibiotic. Okay. Next, question number 25. Which practice reduces disease spread? Option A, avoid clipping seedling tip, excess nitrogen, dense planting, late irrigation. Here the answer is avoid clipping of seedling. As I have already told that the clipping is done on the seedling tip to inoculate the pathogen. So avoiding the clipping of seedling can reduce the disease spread. Next, question number 26. A major resistant donor for bacterial blight or leaf blight of rice breeding is option A, Tetep, option B, TN1, option C, Jaya, and option D, Ratna. Here the answer is Tetep. Next, question number 27. BLB can be used can causes yield loss up to option A 5%, option B 10%, option C 20 to 50%, and option D 100%. So here the answer is 20 to 50%. So generally 20 to 30% it causes, and at the severe condition it may goes up to 50%. Next, question number 28. Hot water treatment of rice seed for management of BLV is done at. So option A 40 to 50 degrees Celsius for 10 minutes. Option B 40 to 45 degrees Celsius for 15 minutes. Then option C 52 to 54 degrees Celsius for 10 minutes. Now option D 54 to 56 degrees Celsius for 15 minutes. So here the answer is it needs to do the hot water treatment for 52 to 54 degrees Celsius for 10 minutes. So this is a seed-borne disease actually, and this pathogen actually resides both on the husk as well as inside the endosperm. So as it is reside inside the endosperm, so hot water treatment is necessary. Apart from this, seed treatment of agromycin is also recommended. Next, the primary source of inoculum is option A seed, option B stubble, option C soil, and option D all of this. Here the answer is all of this. Next, question number 30. The most sustainable approach for BLV management is option A IDM, option B antibiotics alone, option C fungicide alone, and option D deep plowing. Here the answer is IDM. Next, assertion reasoning type question. So first question number 1, assertion: the BLV is a vascular disease, and the reason BLV colonizes xylem tissue. Both are correct. So here the answer is option A. Both A and R are true, and R is the correct explanation of A. Next, assertion: the Kresek is considered as systemic phage, and reason: the pathogen spread internally within the seedling. Yeah, both is correct. This Kresek seedling that is causing the systemic wilt, sudden systemic wilt at seedling stage. Why this systemic wilt is caused? Because of it spread internally through which tissue? Through xylem tissue. Okay. So here also answer is option A. Next, question number 3. Hydrothores are important infection code, and reason are leaf margin are common entry points. Yeah. So here also both A and R are true, and R is correct explanation of A. Because the hydrothores are present on the leaf margin only. That's why leaf margin is the common entry point for BLV. Next, question number 4. Assertion: excess nitrogen reduce BL severity, and reason: Nitrogen suppresses pathogen multiplication. First, this assertion excess nitrogen reduce severity. This is false. And nitrogen suppress pathogen multiplication. This is also false. So both these are false. Next, question number 5. Gene pyramiding improves the durability of resistance. Reason, multiple resistance gene broaden the protection. I have already told. So both this term are true, and R is this A. R gives the correct explanation of A. So the answer is option A. Next, question number 6. Assertion, pathotypes complicate resistance breeding. Reason, different races can overcome specific genes. Yeah, both are true. Because when new pathotypes come, again we have to breed for new resistant variety. So it will complicate the resistance breeding because of the emergence of new pathotypes. And different races can overcome specific gene. Yeah, both are true only. Both are true and it is correct explanation. Next, question number 7. Assertion, TAL effectors contribute to pathogenicity. Reason, they alter the host gene expression. This is already we have discussed. The TAL effector, it is contribute to the pathogenicity. It enhances the diseases. How? By altering the host gene expression. Next, question number 8. Assertion, weather play a major role in epidemics. Reason, rain and wind aid dissemination. This also we have already discussed. So both are correct. The weather play a major role because of the rain and wind, which is causing the secondary spread of the bacterial cells. Next, question number 9. Assertion, avoiding clipping during transplanting helps management of the BLB. The reason, wound facilitate infection. Here also both are correct, and R is correct explanation of A, because this clipping only induce the wound that facilitate the entry of the pathogen. Next, question number 10. Assertion, plants strains in tropical countries are usually more virulent than those in the temperate countries areas like Japan. Reason, tropical climate provide warm, humid environment and continuous rice cultivation that favor the pathogen survival, multiplication and evolution into more aggressive pathogen. Here also both the term are correct, and R is correct explanation of A. Why? Because that in tropical countries more virulent races are found for the BLB. And why? Because it provides warm and humid environment that facilitate the growth and multiplication of pathogen. So in a bigger population, more number of races, pathotypes will be there, that helps them to evolve for evolution and into grow into more aggressive pathotypes. And another important factor is continuous rice cultivation is there for the tropical countries, but in case of temperate areas, in a limited period they grow the rice. Next, the quick revision points. So first it was observed in Japan 1884, then the Kresek phase first reported from Indonesia in 1950, then in India it was first reported in 1959. Causal organism, we have already told, Xanthomonas oryzae, oryzae. That is a gram-negative aerobic non-spore forming capsulated bacterium with monotrichous flagella, and it entered through the hydrothores. Then Kresek phase is systemic seedling wilting that occur in 3 to 4 week plants. Then pathogen colonizes the xylem vascular tissue, that causing the systemic infection. Then primary source of inoculum is seed, soil, crop debris and collateral weed host. The bacterial ooze act as secondary inoculum. Then favorable temperature for the disease development 25 to 30 degrees Celsius. Excessive nitrogen favors the disease. Then resistance donor for the disease are tetep, teducan, Janet, etc. And XA21 is the very important gene. Another recessive gene we have discussed, the XA5. This is a recessive resistance gene for the VLB. Then TAL effector contribute to the virulence. How? By modulating the gene expression of the host. Then IDM or integrated disease management is the most suitable management practice for the management of BLB. So this is all about bacterial leaf blight of rice. I hope this will be helpful for you people. So thank you.